welcome everybody to today's webinar. We're going to discuss today some recent PBPK modelling work that Siri, my colleague Siri, has done to look at how formulating a drug as a salt can give you an advantage in counteracting the DDI that you may get, particularly with a weekly basic drug where gastric pH is altered by a form of acid-reducing agent. There are a significant number of drugs where such DDIs are seen, and it is a significant hot topic at the moment and is required to be looked at by the regulatory bodies. So Siri joined us about 4 years ago. He's a graduate or postgraduate from NIPA in India, the prestigious institute in India. And he's mainly worked on the mechanistic absorption models, as you can see here. So he was being the lead for the expansion of the IVIVC toolkit in SimCip version 20. Where, for example, we recently added the ability to use the diffusion layer model as part of the developing IVIVCs. And he's also spent a significant amount of time working on the virtual bioequivalence module of SimCip that, although we first implemented in version 19, this was significantly expanded and improved for version 21, and Siri is still the lead for the version 22. So this is the upcoming version of SimCip. He's still the lead for the virtual bioequivalence, and he continues to work on some of the mechanistic components of the oral absorption models, such as excipient modeling, for example. So prior to joining us, he spent 5 years as a formulation scientist, working on developing oral drug products, both for new drugs and new drug products, so generics, within Dr. Reddy's laboratories. And we've put Siri's email address here if you wish to contact him. Before Siri goes ahead, this webinar is part of an ongoing series of webinars. This slide indicates the next webinar for which we have a firm date. We're hoping to have a webinar in between this, probably in May, but we don't have a firm date for it yet, so we haven't put this up yet. But as you can see from the slide, the next webinar, scheduled webinar, is June the 22nd from colleagues at Pfizer, looking at OATP1B mediated DDIs and how biomarkers can inform the model-based risk assessment. And as you can see at the bottom, registration for this will be opened soon. But as I said, we do hope to have another webinar in between times. So, having said that, I will now hand you over to Siri to present on the mechanistic PBPK modeling of salts.
Thank you, David. Thanks for the introduction. And first of all, I would like to thank everyone who has joined today for this webinar. And as David mentioned, this is based upon one of our recent publication in Pharmaceutics Journal, where we have studied or where we have used PBPK modeling to predict the advantage of salt forms when dosed with acid-reducing agents. So this is a drug work by colleagues from BMS. So they have developed a drug which is BCS class 2 diprotic base. But as with many of the basic drugs, is when dosed with acid-reducing agents like PPIs or H2 receptor antagonists, they could have a significant reduction in the fraction absorbed or absorption. So even this drug is having a significant reduction in the fraction absorbed. So to mitigate this reduction in the absorption when dosed with acid reducing agents. They have done in vitro salt screening, and as a result of this, they have selected sulfate salt. Uh, so sulfate salt, they have studied the effect of using sulfate salt in preclinical species and found that the salt performs better than the free form when dosed with ARS. So the main objective of this study was to assess if modeling and simulation can predict significant improved performance of salt over the free form when dosed with acid reducing agents. So basically what we have done over here is first we have developed a PBPK model for free form. Once this is developed, we have verified it by predicting the pharmacokinetics of free form when dosed with acid reducing agents like famotidine. So once this is done, we have used the mechanistic salt model and mechanistic surface pH models and two solid state models in SimSip to predict the pharmacokinetics of salt form. So we'll be seeing the details of this in coming slides. Coming to the development of PBPK model for the free form. First, we have used advanced dissolution, absorption, and metabolism model in SimSip. So this is a compartmental transit model where we divide the small intestine into different segments, and we divide in such a way that we can assign different properties to each segment. And as the drug passes from the stomach to different parts of the small intestine or colon, it experiences different environment like pH, bile salts, etc. Due to this, there would be different dissolution in different segments. So we have used an Adam model. Basically, as I said, we divide the small intestine into different segments and assign different properties for overall absorption modeling. And in this specifically, we have used a MECPEF model for predicting the permeability of this drug. So MECPEF model is a model which uses the physical properties of the drug, like log-P, to predict the inherent permeability of the drug through a membrane called P-trans-naught. Once this P-trans-naught is calculated, the model uses the gut physiology, like the dimensions of villi, the plica circularis, and other properties like pH in different segments, or pH in the UBL and UBL layer thickness, etc. So the model uses the physiology of the small intestine and colon to predict what could be the permeability of the drug in different segments of the GI tract. And it also takes into account the paracellular permeability of the drug depending upon the molecular radius of the drug. So we have used MECPEF model to predict the permeability. And then we have used a diffusion layer model to predict the dissolution of the drug. So in SimSip, we use Wang and Flanagan equation or Wang and Flanagan model to predict the dissolution of the drug. And we have parameterized this diffusion layer model using the intrinsic solubility factors, and bile partition coefficients, which are derived by modeling the uh, solubility data in SimSip in vitro toolkit. So you can in SIVA, you can model your measured solubility or measured bioelement media solubility to get your solubility factors and bile micelle partition coefficients. So we have used the measure solubility to get these parameters, and we have used these in the DLM model. Along with these, we have used the measured PSD to model dissolution. So in dissolution model itself, we have two types of models. One is One is mass balance only model, and the other is particle population balance model. So the main difference between these two models is in PPB model, we track the particle numbers in each bin rather than the tracking mass of the drug in each bin. So the way the advantage of this PBB model is it gives more accurate dissolution results than the mass balance only model. So once we have used these models, and then regarding the physiology, we have done all the simulations in healthy volunteers, and in healthy volunteers, we have used a new anatomy model for small intestinal radii and plica circularis values. So this new anatomy model is something which we have implemented in version 20. So what we basically have done over here is the original model, the data, the small intestinal radii and plica circularis values from in the original model was the data was coming before 2006 or somewhere around 2006. But after 2006, there were several literatures where they have measured the small intestinal radii again. So we have collated the newer literature search, and we have updated the small intestinal radii and plica circularis values of human small intestine and large intestine, and we have developed a new anatomy model in version 20. So basically, when users use a new anatomy model, what happens is, because the new radii are quite smaller than the original model radii of the small intestine, it gives more absorption, or the absorption rate itself would be higher if you use the new anatomy. Apart from new anatomy, we have used advanced fluid volume dynamics model. So again, this advanced fluid volume dynamics model is implemented in version 18, which is again based upon new literature data. And when compared to the old model or fluid volume dynamics model, which we had previously, it uses still lower volumes for the stomach, small intestine, and colon, which are more physiological. So once the absorption part is taken care, we have used clinical PK of the drug without acid reducing agents to optimize VSS and clear CLIV of this drug, as you can see here. So once the model is developed for free form using the clinical data without acid reducing agent, we have used this model to predict what could be the effect of acid reducing agent on the PK of the drug. So basically, what we have done is we have changed the uh, pH values for the stomach, duodenum, and jejunum, and along with the CV values. So these are the existing healthy volunteer pH values and CV values for the pH. And these have been changed to 7.2 for the stomach, duodenum, and jejunum, and CV values were reduced quite a lot. And this comes from one of the nice work from Kara, where Kara and her team have studied what is the effect of PPIs and H2 receptor antagonists like famotidine on the pH and other gastric variables. So if you look at the pH in the presence of acid reducing agents, while PPIs could increase the pH of the stomach to around 5 or 5.5, but when we take uh, H2 receptor antagonists like famotidine, the pH is increased quite a lot to about around 7 to 7.2. And also you could see the variability has been reduced quite a lot. About a default CV of 38% to a CV of about 2%. Okay. And well, they have studied this with administration of 240 ml of water, and they have measured the pH at different time points after administration of water. So along with the increase in the gastric pH values, they have also observed that the pH in the upper small intestine has increased as well. So the duodenum and the jejunum 1 pH is around 6 to 6.5. But they have seen an increase in this the upper GI tract duodenum and jejunum pH to around 7 to 7.2 with PPIs and H2 receptor antagonists. So using this data, we have updated the pH values for stomach and duodenum and jejunum, and also the CV values to reproduce the observed pH values in the presence of famotidine. So the other thing which happens when we dose the drug with ARS is change in the buffer capacity in the stomach. Chloride ion concentrations would also change, osmolarity and surface tension would also change. Though buffer capacity is inherent, the change in the buffer capacity with ARS is inherently taken into account by SimSip when we use surface pH models. The change in the chloride ion concentrations are not, and osmolarity and surface tension are not accounted in this particular modeling exercise. And because chloride ion concentrations are important when we dose the drugs as HCl salts, but in this and when we use common ion effect model, but in this case, as we are studying the sulfate salt, we did not take into account what could be the effect of change in chloride ion concentrations in the presence of acid reducing agents. However, if your drug is HCl salt and if you are using common ion effect model in SimSafe, you could be able to take into account what could be the effect of change in chloride ion concentrations in the presence of acid reducing agents. So once we have done this change to the model and we have predicted it, as you can see here, we were able to capture the effect of acid reducing agents, especially famotidine in this case, on the PK of drug. So compared to without famotidine, the observed PK has reduced quite a lot, and we were able to reproduce or capture that effect. So once we have developed the model and verified it by predicting the effect of famotidine on the free form, we went ahead and tried to develop a model for the salt form. So in SimSafe, salt form can be modeled by using few models. One is the mechanistic salt model, and the other important model which we require for modeling salt forms is the two solid state models, which I would come to in few slides. So the mechanistic salt model in SimSafe is composed of three main components. One is the self-buffering effects of the salts. The other one is the salt solubility itself, and the third component is common ion effect. Coming to surface pH. So the surface pH model in SimSafe was implemented way back in version 14, but it was only implemented for acids in version 14. What we have done in version 19 is we have extended this model to bases, ampholytes, and salts. So basically, if your drug is either basic or acid, during dissolution, of the particle, the drug dissolves in the diffusion layer. So in this diffusion layer, because the drug is dissolved, if it is an acid drug, free form of acid drug, it releases hydrogen ions, Okay, because of this releasing of the hydrogen ions during dissolution, what happens is the microenvironment pH on the surface of the particle would be reduced because of these increased hydrogen ions. And this surface pH would be less than the bulk pH because of more concentration of the drug in the diffusion layer when compared to the bulk layer. Similarly, if it is a basic drug, a free form of the basic drug, once it dissolves, it consumes the hydrogen ions in the diffusion layer. Due to this consumption of the hydrogen ions, what happens is the pH on the vicinity or microenvironment of this particle increases. So the pH increases more than the bulk pH because the concentration in the diffusion layer, essentially, when we consider diffusion layer model, would be equal to the solubility of the drug in the diffusion layer. That is the basis of diffusion layer model. Because of the high concentration of the drug in the diffusion layer, there would be increase in the surface pH of the drug when compared to the bulk pH. Because of these, if it is a free form, because if it is an acid, because the surface pH is reduced, it would have lower solubility. And for basic drugs, because the surface pH is increased, it would lead to lower surface solubility of the drugs. Because the dissolution is, this is the dissolution diffusion layer model equation, which we use for calculating the dissolution rate. Because the dissolution rate is dissolution rate depends upon the difference in the solubility on the surface of the drug, and difference between the solubility and the bulk concentration. Because the solubility is reduced on the surface of the drug, the dissolution rate of the drug would be reduced. So basically, if your drug is free form, whether it's basic drugs or acidic drugs, it would have different microenvironment pH during dissolution, and due to this microenvironment pH, the drugs would have lower solubility and lower dissolution than what you would predict using bulk pH. So now, as we see, the surface pH could be different from the bulk pH. But you also have to take into account what would be the effect of buffers on the surface pH. So when we have buffers like phosphate or bicarbonate buffers, what happens is they neutralize this increase or decrease in the hydrogen ions on the surface of the drugs. And because of this buffer neutralizing, the surface pH wouldn't increase as to that extent what we would predict if we do not take into account the effect of buffers. So the surface pH will depend upon the buffer type. It would depend upon the buffer concentration, and it would also depend upon the bulk drug fluid concentrations. And because the buffer concentrations, if we are saying in vivo condition, because the buffer concentrations could differ between different segments of the GI tract, so the surface pH could be different in different segments of the GI tract. And because the bulk fluid concentrations would also be changing during simulations. So due to this, the surface pH which is calculated is not a static pH, but it changes with time. Okay. So that this can be seen in these graphs. So this is the surface pH versus bulk pH graphs in the presence of bicarbonate buffer. So in the absence of bicarbonate buffer, you can see this line, the surface pH is quite different than the bulk pH. But as we increase the bicarbonate buffer concentration, the surface pH would be moving closer and closer towards the bulk pH. Okay. In the similar way, the effect of bulk drug concentration can be seen here. When the bulk drug concentration is 0, which is shown by this yellow line over here, the surface pH is different from the bulk pH. But as we increase the bulk drug concentration, the surface pH would be moving closer and closer to the bulk pH. So in SimSip, we take into account the effect of buffers and effect of bulk drug concentrations on the surface pH and dynamically calculate what could be the surface pH in different segments of the GI tract and at different time points. Coming to in vitro in vivo extrapolation of the effect of surface pH. Usually when we do dissolutions, we study the dissolution in vitro condition using phosphate buffers. So phosphate buffers, because one they are strong buffers, and usually we use higher concentrations like 50 millimolar. What happens is the dissolution in vitro could be different from dissolution in vivo, where we have bicarbonate buffers, which are first they are weak buffers, and the concentration of bicarbonate buffers change between segments, and the concentrations are lower. So we would be needing a model which would take into account the in vivo conditions into account to calculate the surface pH. So in this graph, what you can see is these are the flux. One second. Sorry about that. So these are the flux or intrinsic dissolution rate versus buffer concentration graphs of 4 different drugs: indomethacin, ketoprofen, ibuprofen, and haloperidol. So here, this is the buffer concentration of phosphate buffer. As you can see here, as the buffer concentration is increased, the flux would be increased for these drugs. And using the surface pH model of SimSip, we were able to capture the effect of buffer concentrations on the intrinsic dissolution rate of these 4 drugs. However, when we try to use the same model to predict the effect of bicarbonate buffers on the intrinsic dissolution rate, as you can see on these graphs, we had some problem. So what we did is we have used the measured bicarbonate PK of 6.5 to predict the surface pH of the drug in the presence of bicarbonate buffers for these 4 drugs: indomethacin, ibuprofen, ketoprofen, and haloperidol. And these purple graphs shows the predicted flux or predicted intrinsic dissolution rate. And as you can see here, the predicted intrinsic dissolution rate is higher than the observed intrinsic dissolution rate, which are shown by this dots over here. So the problem was that because we were using the bicarbonate pK of 6.5, we were unable to use our model to predict the surface pH effect of bicarbonate, the effect of bicarbonate buffer on the surface pH. So the reason behind this is because the carbon dioxide reacts with water to form bicarbonate buffers, and this reaction or this hydration of aqueous carbon dioxide with or hydration of this carbon dioxide is the rate limiting step, and due to this being the rate limiting step, we were unable to use PK of 6.5 for of bicarbonate buffer to capture the surface pH. So what we have done is we have optimized the PK of bicarbonate buffer, and using a PK of 4.2, we were able to predict the intrinsic dissolution rate of these 4 drugs, as can be seen from these blue curves. When we use intrinsic when we use PK of 4.2, we were able to capture the intrinsic dissolution rate of 4 drugs in the presence of bicarbonate buffers. So we have updated the simulator in the physiology screen as the bicarbonate buffer effective PK of 4.2. And we use effective PK of 4.2 for bicarbonate buffer to predict the effect of bicarbonate buffers on the surface pH of the drug. Coming to salts, what happens is uh, these act a bit differently than the free forms because when basic drug of salt, say for example HCl salt, is dissolved, it releases the hydrogen ions. Okay, and when acidic drug salt, like sodium salt, is dissolved, it consumes the hydrogen ions. Because of these phenomena, what happens is, say for example, if it is a basic drug, the surface pH of the drug would be lower than the bulk pH. And for acidic drug salt, like sodium salt or calcium salt, the surface pH of the drug would be higher than the bulk pH. Because of this surface pH of the salts, you could see an increase in the solubility and dissolution of the drug. Okay. If you look at this graph, this is the solubility versus pH of haloperidol. These blue boxes, these closed boxes are the solubility of haloperidol HCl. Solubility of haloperidol free form, and these open circles are the solubility of haloperidol HCl salt. As you can see here, so if you look at the solubility itself of salts, you could see sometimes you would not see any significant difference between the solubility of salt versus solubility of free form, as you can see in this graph for haloperidol. But when you use the salt and do and perform dissolution, you would see an increased dissolution of the salt. But when you study its solubility, you would see the solubility is quite less. So this is the dissolution rate or intrinsic dissolution rate of haloperidol versus the bulk fluid pH. Haloperidol HCl salt versus the bulk fluid pH. As you can see here, if this blue line is the predicted dissolution rate when we do not consider the surface pH. And as you can see this point, we are significantly under predicting the dissolution of haloperidol when we are not taking into account the surface pH of haloperidol. But when we see this green curve over here, this is the curve. This is the dissolution rate predicted using the surface pH of haloperidol. So when we are using the surface pH of haloperidol, predicted surface pH of haloperidol, we were able to quite closely match the dissolution rate of haloperidol. So and this is due to the surface pH of this haloperidol salt, HCl salt. As you can see here, uh, at a bulk pH of say 6 or 6.5, the surface pH of haloperidol salt is around 5. So the basic drugs would have an acidic surface pH because of the HCl being released, and this would lead to higher dissolutions. And if we do not consider, or if we do not use the surface pH models, we were unable to capture the dissolution rate of this drug. And when we are using the surface pH models, we were able to capture or we were able to predict the intrinsic dissolution rate of this drug. So basically, surface pH of salts is very important, and this has to be taken into account when we are modeling salts. The other component of the salt model is the salt solubility itself. What happens in a dissolution experiment is when we are performing dissolution, as you can see here, below a certain pH called pH max, the salt solubility would be controlled by the KSP of the drug, or just to say above certain pH called pH max, the salt will have a similar solubility as the free form of the drug. As you can see in this case for haloperidol, above pH max, we above a certain pH called pH max, the salt will have the same solubility as the free form. Okay, so for a salt, there could be a two solubility curves. One is the solubility curve of salt itself, and the other one is the solubility curve of the free base. Okay, and so the pH max is a certain pH above which for bases the salt would convert into free form. Okay, and as you can see here, this is another example of pesopanib HCl pH solubility. So this blue line corresponds to the solubility of the salt, and this green line corresponds to the solubility of the free base. So in a dissolution experiment, when we are using salt to perform, sorry, in a solubility experiments, when you are using salt to perform solubility, what happens is above certain pH called pH max for bases, the salt would convert into free form, or it would precipitate into free form, and then it would use or it would have the solubility of the free form. Okay, and one might say that so this conversion of the salt form into free form could happen in a solubility experiment where we do the experiment for like 24 hours. But what could be the rate or how fast the salt could change into free form above pH max in a dissolution experiment, which happens, the timelines are very quicker. So as you can see, there are two nice studies from Sugano's lab. What they have done is they have studied the effect of, or how quickly the free form will be precipitating from on the surface of the salt form. So in this drug, this is a pioglitazone HCl salt. So within 15 seconds, above pH max, within 15 seconds, they could see the free form of the drug precipitating on the surface of the salt. Okay, during the experiment, and the same happens with diclofenac acid and diclofenac sodium. So because this is an acidic drug, below pH max, very quickly they were able to see that the free form is precipitating on the surface of the salt. Okay, and this is very happening very quickly. So what we do in SimSip is we use the salt solubility until pH max, and after pH max, we use the free form solubility to model the dissolution and absorption of the salt forms. So with the assumption that there would be an instant conversion of the salt form to the free form above pH max for bases and below pH max for acids. And this assumption of instant conversion of the salt form to free form above pH max is based upon these experiments from Sugano's lab, where they see instant conversion of the salt form to free form above pH max. So in Synapse, we use different solubility equations, whether the pH, the bulk pH is above or below the pH max. Okay. So as salt solubility could be different from the free form solubility, and the salt solubility can depend upon the type of drug. Say for example, if it is an acidic drug, it would have different salt solubility. Basic and ampholytes would have different salt solubility and could be defined by different equations. And the salt solubility equations would also depend upon whether the counter-ion is weak or strong, and also whether the stoichiometry is 1:1 or 2:1. So depending upon the drug type, counter-ion type, and stoichiometry, we have derived 16 different solubility equations, and depending upon your drug and counter-ion and stoichiometry, we would use one of these 16 different solubility equations when doing predictions. Okay. And this is the salt KSP model or salt solubility model screen. So one of the important input you have to give is the intrinsic solubility of the drug itself, which is the free form solubility where the drug is not ionized. And the other input which would be needing is the drug solubility at pH max. Okay, and from the drug solubility at pH max, we would back calculate what is the KSP. So KSP is nothing but the equilibrium constant of salt dissolution. So at pH max, KSP would be nothing but the uh, would be nothing but the salt solubility into salt solubility, or the square of the salt solubility. Okay, and when you define and the user have to define what type of counter-ion it is. In this particular case study, we have used intrinsic solubility of the drug. We have used the drug solubility at pH max, and we have used dichroic strong acid as the counter-ion type, and sulfuric acid as the counter-ion. So when you select these, the properties of the counter-ion would be populated in this screen, and these properties of the counter-ion are required in calculation of surface pH of the drug or of the salt. And as I said, we use different solubility equations depending upon if the drug is acid, base, or depending upon counter type. These are two examples. So atorvastatin calcium. So we have a equation, different equation for the salt and different equation for the free form. But the final equation or the final solubility which we use in the simulations would depend upon what is the pH max. So below pH max, we would use the free form solubility, and above pH max, we would use the salt form solubility. Similarly, for this is an example for zolpidem hemitartrate. This purple line is the solubility of the salt, and this blue line is the solubility of the free form. So above pH max, we use the free form solubility, and below pH max, we use the salt form solubility. So in the simulation, you need not mention what is the pH max. Depending upon the solubility curves, we would calculate what is the pH max. The only inputs which are required for modeling are the salt solubility at the pH max and the intrinsic solubility of the drug. So the third part of the salt model is the counter-ion effect model. For some drugs, specifically chloride salts, what you can see is reduced dissolution when the pH is reduced. And this is due to the common ion effect. So this common ion effect, because as you know, the KSP is a equilibrium constant, and if chloride ion concentration increases, because KSP is a constant, the drug concentration decreases. Because the drug concentration decreases, the solubility decreases. And this common ion effect can be modeled in SimSip, and we have collated the concentrations of the common ions like chloride, sodium, phosphate, and calcium in different segments of the GI tract, and these are available in the population screen. And the user has to use the endogenous ion model uh, to take into account what is the effect of these counter ions or common ions on the solubility and dissolution of the drug. And these endogenous ion concentrations are not static, and they are dynamic in that they are diluted with water when we take 250 ml of water. So these are the examples of the simulations. This is the model predicted chloride ion concentration in stomach, and model predicted chloride ion concentration in the duodenum, and these blue dots are observed chloride ion concentrations in stomach and duodenum. So we use a dynamic endogenous ion model, and using this model, you can model common ion effect of your drug. So coming back to the case study, when we have used the salt model and the surface pH model of the salt, and try to predict what is the PK of the salt form when dosed with famotidine and when not dosed with famotidine. So this gray curve is the PK of the drug of the salt form when without famotidine. As you can see here, there is no significant change of the dissolution of the PK of the salt form when compared to the free form, which is this blue curve without famotidine. But if you see this green curve, which is the salt form, when dosed with famotidine, and this orange curve is the free form when dosed with famotidine. What we were predicting is very higher concentrations when compared to the free form when dosed with famotidine. And these higher concentrations or higher exposure when the drug is dosed as a salt form along with famotidine is in line with what has been observed in preclinical species beagle dogs. So in beagle dogs, so unfortunately the study has not been done by BMS in the humans, but they have done the study in preclinical species. So what they observed is when salt form is dosed along with famotidine, they could see higher exposure when compared to the free form when dosed with famotidine. So the same observations or the what we are predicting is in line with the observations in preclinical species. So we could able to say that the modeling helps in this case to predict the advantage of salt form when dosed with acid reducing agents. So again, the reason behind this increased the reason behind this increased absorption of salt form when dosed with famotidine is due to the surface pH. If you look at these two graphs, the green curve over this green curve over here is the bulk pH, which is around 7.2 in the presence of famotidine. But if you see this red curve or the orange curve, which is the surface pH of the salt in the presence of famotidine. As you can see here, even the bulk pH is very high in stomach, and also in duodenum. Similarly, in other segments of the GI tract, even the bulk pH is very high in the presence of famotidine. The surface pH is very low, and due to this lower surface pH, what happens is there would be higher surface solubility as seen in this graph. So this one, this orange curves are the surface solubility, and these green curves are the bulk solubility. In stomach and duodenum, you could see that there is higher soluble surface solubility when compared to this bulk solubility when we dose the drug as a salt form with famotidine. Due to this higher surface solubility, we were seeing higher absorption of the drug. However, when we do not use the surface pH model, you could see here the green curve, which is the salt PK with famotidine without surface pH model. There is no significant improvement when compared to the free form in the presence of famotidine. So it is very important, at least from this study, we could see that it is very important to consider the surface pH of the salts when trying to model them. So another important thing to consider here is, though we are seeing an increased absorption of the drug when dosed as a salt with famotidine, still it is lower than without famotidine. Okay, so without famotidine, the pH is say 1.5, and with famotidine surface pH is around 2. But still we are seeing a significant lower absorption of the salt form with famotidine when compared to the free form without famotidine. So the reason behind this lower absorption of the salt form with famotidine when compared to the free form without famotidine is due to the precipitation. So what was happening is, so because as I said previously, because the surface solubility is very high for the salt when compared to the bulk solubility, it leads to supersaturation in the stomach. So these are the concentrations of the drug in the stomach, this orange graph, and this green graph is the bulk solubility of the drug in the stomach in the presence of famotidine and pH 2. As you can see here, because of this higher surface pH and higher solubility, you could see a supersaturation of the drug, or the concentration of the drug is very much higher than the bulk solubility. And because the concentration is very much higher than the bulk solubility, what would eventually happen is it would precipitate out. So the salt form on dissolution, it goes into the solution. So what could happen is it could either precipitate back into the salt form, okay, because of the supersaturation, or it could precipitate into the free form, okay. And in this particular case, it's difficult to precipitate back into the salt form because precipitating back into the salt form would require the counter ion concentration to be very higher. Uh, I would come to that in next slide, but would require counter ion concentration to be very high. But what happens in this case is because the counter ion, which is sulfate in this case, would be diluted with GI tract fluid volume, the concentration of the counter ion would be lower uh, or would be insufficient for the drug to precipitate back into the salt form. So what happens is the drug solution would precipitate into the free form, okay, And how we model this is in SimSafe is by using two solid state models. So one of the advantage of using population particle balance model is we can able to use two solid state models. So how we model in this case is because the drug which is given in the salt form goes into the stomach and then precipitates as a free form. We can use the two solid state model in SimSafe, and in the dose fraction in the dose for the solid state one, which is the salt form, is set as 100%, and in the dose fraction for the free form is set as 0%. And in the precipitation model, we have selected precipitation to solid state 2, which is defined as the free form. So because we were selecting the precipitation to solid state 2, the drug would precipitate as a free form. And as you can see in this graph, this is the solid state 2, which is the free form of the drug in the stomach at different time points. So as you can see here, initially the free form amount in the stomach would be 0, but as the drug is precipitating, the free form, the amount of the drug of the free form, because the drug would be precipitating, is increasing over time, and then it's decreasing due to transit and again redissolution of the free form. Now, because of this precipitation of the drug as a free form, and then this free form would dissolve using the free form surface pH and free form solubility, the dissolution would be slower. So that's the reason why we are not seeing a very significant increase in the salt form absorption or salt form AUC when compared to the free form without famotidine, but still better than the free form in the presence of famotidine. So what would happen if we do not use free form as a solid state 2, and just let the simulator precipitate the drug as a salt form. So as you can see here, this is the graph or the predicted salt form concentrations. Okay, predicted salt form concentrations with famotidine when we do not use solid state 2, or when we do not use or we do not tell the model that the drug would precipitate into a free form. So as you can see here, the concentrations predicted for the salt forms are very much higher than what we have seen in the preclinical species. So it is not in line, the predictions are not in line with what we have seen in the preclinical species. So it is really important to take both into effect the salt model, the salt surface pH, and also the fact that once the salt dissolves in the GI tract, it might not precipitate back into the salt form, but it would precipitate back into the free form, and because of this, there would be increase in the absorption of the drug, but not to the level that could be seen with the free form without famotidine. Again, just to reiterate why the drug could not precipitate back into the salt form. So how the model works is the drug could precipitate back into the salt form, but only when the product of drug and counterion concentrations are greater than the KSP of the salt. So however, because this counterion is being diluted, sulfate in this case is being diluted with the GI tract fluids, the product of the drug concentration and counterion concentration would not be above KSP for the drug to precipitate as a salt. As you can see in this graph, without using solid state 2, only using the model, telling the model to precipitate as a salt form. As you can see here, it doesn't precipitate back into the salt form. This is the cumulative fraction. Sorry, I guess. This is the cumulative fraction dissolved, and this is the cumulative fraction absorbed. As you can see here, there is no precipitation of the drug back into the salt form, even when I use CSR of 1 and PRC of 1000. So in conclusion, in this particular case study, what we have done is developed a PBPK model for free form and predicted the effect of acid reducing agents on free form. And we have extended this model to predict the dissolution of salt form using mechanistic model. So what we can, at least from this study, what we can say is PBPK can be used to predict the dissolution and absorption of the salt forms. However, we would be needing more case studies. We would be needing to work on more case studies on this aspect. And critical to this these PBPK modeling predicting the dissolution of salt forms is the mechanistic surface pH and two solid state models. Once similar kind of models are developed, these can be further used in screening of the salt forms and also could be used in studying the effect of salt disproportionation during storage and its effect on PK. Thanks for your time, and I would be happy to take any questions. Thank you.
Thanks, Siri. Obviously, we've run over a few minutes, but hopefully people can hang on for a few questions. I have answered some of these in the chat, so given that we've overrun, I'll select some of the later questions that I haven't fully answered. So there's a question on how the precipitation model was parameterized in this case study.
In this case study, we have used default. In this particular case study, we have used default precipitation parameters. We did not modify them because we were able to, as you can see in this graph, we were able to predict the PK of the drug with the default precipitation parameters. Ideally, we would have given the data, if we would have the data, we would have modeled in SIVA to get the precipitation parameters and model. But in this particular case, we have used the default values because they were working all right, as in this graph and also in the verification of the model.
Yeah, so we've underpredicted Tmax somewhat, but Cmax is not too bad.
Yep.
There's a question about the pH in the presence or after the stomach pH as a result of the impact of famotidine. So we set this to 7.2 based on Kara Litu's data. So the question is, was sensitivity analysis done on this pH? And also, what is the mean stomach pH in the simulated virtual population?
I mean, the sensitivity analysis particularly was not undertaken in this study in this case, but as you can see from the results, the dissolution is quite sensitive to the change in the pH of the stomach and upper small intestine. As you can see here, the concentrations are or the PK is quite low when we change the pH. So it's quite sensitive. It's evident from there, and given the the CV is very low, and again coming from the data where you can see the the distribution of the pH is not quite variable. So the mean pH of the simulated individuals would be quite closer to 7.2.
Yes, but each subject gets assigned a stomach pH based on the mean and the CV, so sampling of a tight distribution. And it's interesting to note that we have this very low CV on the gastric pH after famotidine, and if you look at the clinical study, you can see the plasma inter-subject variability is very much reduced compared to without famotidine. So it may be there may be a link or a relationship there.
CV over here.
There's another question. Typically, companies try to avoid pH-mediated DDI effects and use enabled formulations. If we have data where a salt does not show PPI-ARA effect, has such a model been used as part of the validation? And a comment, I guess, also a prediction of a no ARA effect is also important. Yes, I think we agree with that.
Yeah. So I mean, in this particular case, there is some ARA effect for salt as well, as you can see here, but it is not quite significant as we see for the free form. So there could be cases that there could not be, there wouldn't be any ARA with the salt form. Yeah, case to case basis. So the basic modeling strategy or the models can be used to predict similar kind of observations.
Yeah, I think
David,
the
you want to
question
add
is,
something?
I think the question is, do we have an example of a salt which does not show this acid reduction effect? Which we don't at present.
No.
So but we take your point. So I guess in principle the models could be used to screen different salts, and some will have this surface enhancing insolubility effect and some may not, or it may not be significant and affect the PK. The question is, does anyone then proceed with such a compound based on their in vitro screening through to the clinic to prove with the aim to prove that the PBPK modelling could also predict no effect? The answer is I don't know. Related is adding acidic excipients to overcome the PPI effect in addition to salts could be a formulation fix. Does the model account for the addition of acidic excipients?
Yeah, I mean, in the present SimSip models, what you can use is a user input surface pH. Let me see if I have that screen. So yeah, I mean, it is one of the strategy to use acidifying or basifying agents to as a formulation strategy. But in this case, because these acidifying and basifying agents would dissolve in during the dissolution would dissolve, and then it's a complex process, and the concentration of these acidifying or basifying excipients would be reduced due to the dilution effect. It's difficult to mechanistically predict what could be the effect of the pH, or what could be the effect of those excipients on the mechanistic surface, on the surface pH. But what you can do is you can use user defined option in SimCip. So the when you use this user defined option, what you can do is you can take your formulation and crush it and measure the microenvironment pH in the presence of the excipients which you are using in the formulation, and measure the microenvironment pH, use it as user defined surface pH in SimSafe, and then you can predict what is the effect of those excipients on the PK of the drug. So basically, at this moment, we do not have a mechanistic model to account for the acidifying or basifying excipients, but we have a user-defined model where you can input your surface pH of your formulation to see what is the effect.
And you could also use the model the other way around and look at, well, what surface pH would I need to aim for to get the PK profile that or dissolution and PK profile that I want.
Yes.
Masood has pointed out that there's a wish list item for version 23 on pH-mediated DDI. So we will look at more case studies as a part of that project, and we would like, as usual, consortium members to share data or case studies that they have or prepared to share. So I think we can draw this to a close now. If any of you have any further questions, Siri has put up our contact emails here, so please contact us, and please share any please share any data you have on gastric pH mediated DDIs, as I already mentioned. So thank you very much for your attention, and goodbye to everyone, and thank you very much for your presentation, Siri.
Thank you everyone. Thanks. Bye.